Welcome to Biotech Stage, a stage where we talk with biotech professionals from the whole world and from the whole spectrum of job functions, whether it is president, vice president, entrepreneurs, directors, team leaders, and most importantly, the people who work on bench and work on day-to-day experiments. That means scientists. Today, our guest is Shawn Smith. Shawn has been in the industry for almost 3 decades. He is an expert when it comes to cell culture, process development, business development, and all the related expertise that are needed for successful commercialization of biotech drugs or biotech maps or biotech recombinant proteins. He is one of the early member of cell culture bioprocessing community. He has worked in companies such as Thermo Fisher, GE, Fujifilm, MilliporeSigma, Paul, and ZeppoMatrix. Welcome, Shawn, on this stage.
Well, thank you for inviting me. Yes, it's been quite a journey for me. You mentioned a number of companies that I've worked for. Well, they've all changed names over the years. So I may mention names of companies or roles I've had within different organizations that might seem a little different, but I'll try to always bring it back to a common root so that people understand. But yes, my journey started over 30 years ago, and even though I'm technically retired now, I still feel like I'm on that journey. So I think one of the key learnings for everybody that's listening in today is to have a lifetime of passion for this field in which we're working. So biotech, life sciences, biopharm, whatever you want to refer to it as, having a sincere passion for learning at all times is, I think, the single most important success factor. So yes, I started off as a bench scientist. I have an undergraduate degree in the sciences, so I'm kind of a rare entity in that I did not follow through with a PhD. I do have some regrets with that. I love the science, but I am not an academician in nature. So I wanted to get hands-on in the laboratory, start working basically as a chemist in a basic research setting. And from there, my journey continued onward and upward.
So thank you very much, Shawn, for such an important lesson you already gave. One of the important thing that people need to remember: lifetime passion for biotechnology. So I wanted to ask you: you started as a bench level scientist, and then you grew to president and CEO. How did you manage such a big change in both role and functions, and the level of responsibility? Obviously, it's a big change. Bench level scientist has certain other responsibilities, and CEO has a different level of responsibility. So how did you manage such a change?
Wouldn't it be great if I could just put together a simple roadmap to describe that journey and others could embark on it? It was a very long journey, so such an evolution does indeed take a career. And with that, I'd say it's kind of a combination of patience and perseverance. So anybody starting off, regardless of the level of education you have, whether it's bachelor's degree or PhD, some level of patience is required. So during your formative or early years, it's learning. It's all about learning. You need to drink in the information and continue to grow in your understanding of what you're responsible for, what your company's doing, and what the state of the science is driving towards. So it's always staying on top of things and moving as the science moves, again, understanding what your company expects from you and doing the best you can. So it's a sincere level of effort, but it's also, again, as I say, realizing that things do take some time. But in the course of being patient, it's also important to persevere. And persevere means overcoming challenges, staying with your, let's call it, determination or ambition to continue to drive towards higher levels of responsibility. And that's the bottom line. So when one changes positions from bench level to supervisor, manager, director, and above, it's always about assuming higher levels of responsibility within the company. And that means making decisions that have a bigger impact on the performance of the company. Those aren't always easy things. It does cause stress or pressure, and that's not for everybody. So part of embarking on that journey again is I'd say the courage to assume that type of responsibility and occasionally getting it wrong. And well, you may hear from others that, failure is part of success and learning how to fail and recover and move forward is always important. I mean, there's truth to that. I mean, you got to be right more often than wrong. But in any event, it's the ability to say, I can do it. I've got the confidence. I've got the information around me. Even if I don't know it all myself, I have it within my grasp to learn it. And it's that, again, passion to continue learning throughout your career. And every step of the way, never slow down with that passion. Because if you think you learned everything, that's when you start to fail for real. So you continue to drive forward with learning, understanding, and it's not just business systems. So becoming a senior manager in business isn't just about the numbers. It's not just about understanding the organizational chart. It's understanding the people you're working with, what motivates them, what they care about, and sharing in that kind of exuding the passion that you have so that others also embrace that and collectively as a team. And I love making sports analogies when it comes to business or the working environment, but it truly is a sports analogy, regardless of the sport, if it's cricket or baseball or a different type of a world sport. It's the camaraderie of the team. It's leadership more than management. So it's not barking out orders in a hierarchical chain of command. It's more about the collectivism of working as a team and understanding that we all have different roles to play. And in measuring those roles against the performance objectives, because you have to know if you're on the path to success or not. So, we come back to business planning for life as well as within a company or within one's career. You have to have objectives. You have to shoot for something in terms of ambition. Beyond the objectives, you set a strategy that says, this is how I'm going to go about achieving those objectives. You fill in the details, which are called tactics, and around them, you have measurement systems or metrics. You hear all this. I know you hear this in management meetings. People are telling you that these are important factors in running a business, but I believe they're important factors in running your life as well as your career.
Yeah. Thank you, Shawn. These are very motivational and inspirational words. And I'm sure the people who are listening and people who will listen this to, and they are working on bench as an entry-level scientist or managers or directors, and they want to go to another higher position, these will definitely help them a lot. So, Shawn, one other thing that I wanted to always ask you, during this time, I'm sure you paid careful attention to certain things and you avoided certain other things. So, what do you suggest someone who is in similar position and trying to move from R&D to sales or marketing or the management job?
Well, I'll say one thing that might be provocative. It's really not about the science. It's not about the business structure. It's about the people. So you're going to deal with people throughout your career. Sometimes that's referred to almost negatively as politics. But when there's human to human interaction, there's politics, not governmental politics, but office politics or company politics. And understanding how to navigate that situation, that environment is critical. So personally, well, I guess what's important to understand is everybody's a little bit different. I mean, humans are humans, but people have their own personalities, and you can't control how others behave. You might be able to influence them. So I found that it's absolutely critical for me to operate off of a platform of mutual respect. Now, wouldn't that be great if everybody behaved that way? Everybody respected everyone else's opinion and we all worked happily. That's not the way reality is in most cases. So you have to understand that sometimes you need to earn the respect of others, and other times it might be difficult to give respect to others who maybe rub you the wrong way. So understanding how to work with others, but starting with that initial attitude of mutual respect or reciprocating levels of respect, I think are very important, again, at every level. And I think it's important to look somebody in the eye and be genuine with them and be honest with them. And that's the first step of earning respect, is to be able to connect with people at all levels. There are hierarchies in business. You will have a CEO, you will have an entry-level person that just came out of school, and they have different roles and responsibilities within the company, no doubt. Different levels of accountability that have impact on the business. But regardless of the level, treating people with respect is key, and I have found that to be helpful. And it's not always easy. And again, sometimes it's harder to give respect to somebody whose behavior you don't favor. You see them maybe treating people in a way that you don't agree with, and it can be frustrating. again, you do your best to maintain composure, to work with that person, to help steer them towards a more palatable behavior with others, but you can't necessarily control it. I just think that, operating off of that type of again, a platform of mutual respect is absolutely key.
Yeah, thank you. Respect is something that is obviously people understand. It's very important. However, in our daily life, we sometimes forget how to gain the respect by giving the respect.
That's
So
right.
this is very important for people to remember. So Shawn, my one other question that always wanted to ask you: you have been in this industry for almost 3 decades, and a lot of things have changed. I mean, the way the science was being done in 1990s is very different from the way it is being done nowadays. For example, initially back in 1990s or 2000, there was a focus on animal component free media. I mean, when we are talking about recombinant proteins or MAPs, and that was a big thing. And now slowly it was changed to you need to increase the yield from 1 gram per liter to 10 grams per liter or even more. And now there is a focus on cell and gene therapy. So what do you think will change in next few years, or how it is shaping up the biotech industry?
Right. Well, I can say that I started off in the cell culture arena back when classical media supplemented with serum was the standard. So in the late 1980s, I was
Yeah,
there for
DMEM plus 10% serum.
exactly. So my R&D efforts were involved with understanding precipitates in classical media, trying to figure out what made serum work, things like that.
Hmm.
And then I was there for the first generation of serum-free media. And as you noted, the progression from serum-free to protein-free to completely animal-free and ideally chemically defined, which are, discrete molecules. So what we witnessed there were few things happening. And I think that these elements continue today. They've been going on for a long time and they'll continue to go on. And that's really the combination of regulatory compliance. How can we reduce risk? Sounds simple, but looking at potentially contaminating components such as animal origin materials that might bring a virus or a prion into the medium is a problem. So eliminate that. That's a compliance aspect. It's reproducibility or consistency in processing. That's always a challenge in biological systems. It's been the bane of cell culture from its beginnings. Doing small scale research, biological research in a petri dish or a flask is one thing, but scaling up to a 10,000 liter bioreactor and having the exact same performance time after time is a different type of challenge. So you got regulatory, you've got performance, and then of course you've got the economics of the system, the productivity, the cost basis of the material being produced. And that's comprehensive, not just the upstream part of cell culture, but also downstream with purification, which also, as we know, adds a lot to cost. So I think these three factors will continue to evolve. There will always be pressure on executing against all three. You can't sacrifice regulatory compliance to gain more productivity. People might try, but there will be other parts, checks and balances within the organization, like regulatory affairs or QA will say, that sounds good, but time out, we need to characterize, we need to validate, et cetera, et cetera. So I think, again, I talk often philosophically about things, but maybe it's a cultural issue. I feel that in the early 90s, cell culture and biotech in general was a lot of biology that veered on art, and it's transformed into more engineering, biological engineering, and it's become much more, I'll say, math oriented.
Hmm.
Less art, more hard science, again, more engineering. And I think there's a fundamental difference between a biologist, scientist, and an engineer. Different ways of thinking about things, different approaches to what happens in the laboratory. The cell culture scientist, the biologist cares about, the cell cycle a little bit more and what the phase of the moon might be, whereas the engineer is really looking at numbers and off-gassing and stuff like that. So let's call it the discipline of the science, the migration to higher levels of detail within that science is going to continue. And that's just cell culture bioprocessing. And by that, I mean the antibodies and the recombinant proteins that are made at very large scale today, blockbuster drugs throughout the world, biosimilars in many cases have been very successful, certainly in India as well as other countries. In terms of other techniques involving cells, such as cell and gene therapy, and where that's heading, it's been occurring now for many, many years. I certainly have worked in it in my time in the field, but I think we've got a long way to go there. And part of it is really helping to educate society, the general public. I mean, I still think there's a lot of fear and trepidation about the unknown. Sometimes that is unfortunately political and in the other sense of the word political, but people don't understand it. They don't accept it. It becomes more challenging to push the science through into the world of therapeutics. And again, when it comes to the science or the characterization of that science to gain the regulatory approvals, it really is about that discipline of characterization, better understanding, depth of understanding, so that reproducibility can be more assured. I mean, there's going to be some variability because it's biological, but the more reproducibility, I think the greater the collective confidence in these technologies.
So, Shawn, thank you very much for your feedback on how industry has changed in past 10, 15 years and your 3 points about what companies need to look for in coming years or so. And those 3 points are very important: the regulatory point, reproducibility of proteins, and the economics. So, Shawn, now I want to focus on something that is also very important for companies, and that is the cost of the final product. So, whether it is a MAB or whether it is a cell and gene therapy product. So, what do you think the cost be dependent upon? The cost of raw materials or the process efficiency? What do you think will bring the cost?
Right. So, of course, the answer is both. Obviously, raw materials play a key role in understanding those raw material contributions are, of course, important. But I think that what most companies have found is that the actual raw material contribution to final cost of goods sold is relatively minor compared to all of the other costs. And that's a big bucket or big concept to contemplate. And the other costs are yeah, you can call it process efficiency, but what does that really mean? That means time, that means yield, that means scrap or loss during processing, which was part of the yield factor. But the cost of the actual raw materials, be they upstream or downstream, or even some of the perhaps single-use components or filters or whatnot that are involved throughout the processing, those certainly contribute. But if we think about labor, and not just manufacturing labor, but the supporting infrastructure, what is often referred to as overhead, such as QA, regulatory, maintenance, engineering, the people that keep the bioreactors going, the people that move the raw materials around the facility, all of those costs, including the environmental controls and management of that facility, add up to the total cost of goods sold. And those costs can be fairly significant, as we know, which is why quite often biotech drugs, at least initially, are quite expensive. So the focus on cost cannot be unidirectional. It has to be comprehensive. Yes, raw materials matter. They can't be neglected. But in terms of what has the greatest impact on cost, it is really that holistic picture, which involves the process, the validation of the process, and the monitoring of that process. So those various costs coming together are, maybe best described as efficiencies and process effectiveness, but that's going to have a bigger impact than any chemical reagent or even the chromatography resins, which are often the biggest part of the cost picture.
Okay, thank you, Shawn. Yeah, it is important to know process efficiency is, as you said, is probably more important than cost of the raw materials.
Yeah.
Efficiency is something that can always be improved.
And there's trade-offs in that there will be times, quite likely, where a more expensive raw material might contribute to higher productivity. So then it becomes a matter of maths, right? I mean, one has to calculate, does it make sense to incorporate the more expensive component to improve your overall yield factor? But what could interrupt that process, if you will, in terms of implementing a change such as that, will be the characterization or validation. So, there are times when a newer or more, let's call it value-adding raw material could contribute to greater process efficiency. But if it's not well characterized, if it's that proverbial black box, then it might be a non-starter.
Okay.
So part of what needs to occur, obviously, is a kind of a collaborative interaction between that raw material supplier as well as the user, so that they understand what they're dealing with and can properly integrate it into the process.
Okay, Sean. So now let's just focus on something that can control the process efficiency, and that is the people who work there. So I want to get some of your feedback on what do you advise or coach for the younger generation? What they need to focus on, obviously for their own growth and for the process efficiency.
Right. So I've mentioned earlier the need for kind of a lifelong passion or commitment towards this industry. I mean, that's got to come from within. I mean, really, again, it's that kind of inner desire to keep learning about what's happening in your field, and that to me is such a critical message to the younger employee, and that's don't stop learning. So
Mhm, mhm,
what you're doing today is probably not what you're going to be doing in 5, 10, 15 years from now. So continuing to learn about the nature of your organization and really kind of move a bit tangentially if possible. And what I mean by that is learn about the adjacencies within your department, your company, so that you are able to learn again about more comprehensive roles that the organization has. So maybe today you're working in one particular department or area. Learn what somebody's doing just upstream or downstream of that so that you
mhm,
can gain an appreciation, and that will help round you out, if you will, in terms of your
mhm,
skill set, your knowledge. The more you understand about the different aspects of research through manufacturing to ultimately commercialization, the more value you will bring to your organization. And that's a very broad statement for me to make. But a lot of it depends on, again, what the individual desires or what they find illuminating or gratifying in their own pursuits.
mhm,
So, an example of that might be intellectual property law. So,
mhm,
does a researcher today, perhaps with a bachelor's degree in science, have interest in patent law? Are they actually interested in understanding what makes a technology or a product more
mhm,
unique and therefore able to be patented? Do they want to get into working with contracts, licensing, royalty schemes, etc.? And I bring up intellectual property law as just one example that's a diversion from working in a laboratory as a scientist. But what I find in the industry is the best intellectual property attorneys start off as scientists. They understand the
oh, mhm,
science and then
mhm,
they learn the legal aspects of IP, and then they kind of migrate or evolve into that role. And that can be a very senior role. It certainly represents career progression and
mhm,
differentiation of that career. You're no longer just in one area. You've now broadened your scope of
mhm,
responsibility. And again, that's legal, that's administrative, that's one particular area. Again, certainly not for everybody. But some folks might say, listen, I've worked in R&D. I understand how to conduct certain experiments, run certain protocols. I now want to see what is the next step in delivering a drug product to the market. I want to work
mhm,
in manufacturing. I want to
mhm,
take the basic knowledge and then apply it into, again, more of an engineering type of environment, so that we talk about scale up,
mhm,
productivity, and again, cost management and all that. But again, I think the key is not staying in one spot. It's having
mhm,
the courage to try new things, to explore. And part of that exploration isn't just about conquering the challenge of doing the activity. It's
mhm,
understanding, again, within yourself if it's something you enjoy doing. Because if you move into an area, you give it a try. Let's say I want to schedule production. I want to be a planner or scheduler because you think that that's of interest to you. You try it and you're doing it for 3 months, 6 months. You realize I don't enjoy doing this. I'm not motivated. This does not give me a reason to wake up and go to work in the morning. If that's the
mhm,
case, you don't want to be in that area for too long because you're gonna become disillusioned and then perhaps not as good of an employee. So
mhm,
it's that balance of learning and willing to learn new things, but also internal or introspection to understand, what is it that motivates you. So it's
mhm.
that constant kind of external internal reflection that I think is key. And again, that's big stuff to say in terms of more philosophy than business planning, but I really do believe that's critical. And I don't think that people should ever stop thinking that way. Every stage of their career, they need to be thinking about what interests them, what motivates them, and what can they do to further their career in that area by learning new things and experiencing new things.
Okay, Sean, thank you. I think this will be very helpful for all the people who are entering into the biotech field and trying to stay motivated, trying to move up into the some different roles and some senior roles. Okay, so now we are coming towards the end. And I want to finish this conversation or the podcast in two sections. In first one, I request you to share a particular story, particular funny story, or during your career, or if you want to share something strange thing about a biotechnology that people, general public, are not aware of. And in the second part, we will do the rapid fire questions of a series of 10 questions. So, Shawn, if you could please either share a funny story or just share something strange about biotechnology.
Okay, so maybe not a technical or scientific issue, but one thing that I've found kind of amusing along my journey is the use of acronyms in our industry. So we all live in a world of acronyms, they're all over the place. But as it relates to cell culture, there's a couple that actually speak to my hometown. So I'm from a place in the US called Western New York. Most people in the world have heard of Niagara Falls, but not so much Buffalo, perhaps. You hear New York, you think New York City. Very far, very different from New York City. But the first major role I had in this industry was at Gibco. Now, Gibco has been part of Life-Tech and Vitrogen, Thermo Fisher, but Gibco itself is actually an acronym. And it stands for Grand Island Biological Company. Grand Island is an island in the Niagara River, right before Niagara Falls. So someday it may break off and go over the falls. But it is actually a town in Western New York, which gave rise to this cell culture company. One of the flagship products of this company, at least in its early days, was the classical medium RPMI 1640, which also speaks to my hometown. RPMI is an acronym for Roswell Park Memorial Institute. So it was a classical basic medium to which serum was supplemented, commonly used in the study of cancer cells. And that originated at Roswell Park, which is also part of our local community. So these are just acronyms that, unless you're kind of a cell culture science nerd, you're not even going to be very familiar with. But if you are in the cell culture industry, you've certainly heard these acronyms, and now hopefully you know where they're coming from.
Okay, thank you. This will be, I think, very funny, and some people will find it very interesting. Thank you. So, okay, Shawn, now it's time for rapid fire questions. I'm going to ask you 10 questions, and you need to give your response on a scale of 1 to 10, 10 being the best, almost there, or strongly agree, depending upon the type of question. So, first question: quality of biotech drugs in India.
9.
Production or capacity in India.
8.
Innovation in India.
9.
IP protection in India.
7.
Adequate availability of workforce in India.
9.
Productive interactions among the peers.
7.
Capital available, public or private, for new research.
Not sure.
Cost efficiency with an aim to reduce cost of drugs.
8.
Unambiguous guidelines, regulatory process.
7.
And finally, question number 10, make in India efforts.
4.
Okay. Thank you, Shawn. That's the end of the rapid fire questions. So again, Shawn, thank you very much for coming to the stage or studio today. And it was great talking with you, and so much of learning, so much of feedback, so much of tips. So thanks again.
Yeah, you're very welcome. I appreciate the invitation. Looking forward to doing it again.
Okay. Thank you again. Bye-bye.